Scientists have unearthed DNA fragments that are believed to be over 2 million years old. Experts say the crucial discovery from Ice Age sediments in northern Greenland opens a new chapter in paleontology.
Deep in the frozen north, scientists are cutting into ice that hasn't seen sunlight in millions of years. But they're not searching for fossils or gold. They're collecting soil. Inside those frozen grains, they uncovered something that completely rewrote what we know about life on Earth. And the real shock? Some of what's buried down there isn't dead. The world's coldest treasure hunt. Far up in northern Greenland, just a few hundred miles from the North Pole, scientists have been carving blocks of permafrost from a place called the Cap Kubenhavn Formation. It's not just ice or dirt, it's a time capsule. Every grain of that frozen soil is like a letter from a world that vanished long ago. To collect it, researchers from the University of Copenhagen go out onto the tundra during the short Arctic summer, when daylight lasts around the clock, but temperatures still hover near freezing. They find a clean face of the exposed ground, pale, smooth, and wind-polished. Then scrape away the top layer that's been weathered by time. Once the fresh surface is ready, they use a diamond-tipped drill to carve out a perfect cylinder about 4 inches wide. Or sometimes they cut a solid block about 16 inches across. It's a delicate operation. They can't let the drill heat up or melt the ice, because even a small thaw could destroy fragile traces of DNA locked in the soil. When they finally lift that frozen block, they treat it like treasure. The sample is slid straight into a lined metal box and sealed inside a portable freezer. From the moment it's cut to the moment it's back in Copenhagen, it never rises above -8 degrees Fahrenheit. Everything, the tools, the gloves, the air around it, has to stay clean and cold. If it ever thaws or gets contaminated, the ancient molecules they're hunting could disappear forever. Over 3 separate field seasons, in 2006, 2012, and 2016, the teams repeated this same precise process at 5 different spots along that Arctic outcrop. Each sample was logged, labeled, and packed with surgical care. They always cut deeper than the outer crust, taking only the untouched heart of the permafrost. This kind of soil harvest isn't just happening in Greenland. Across the Arctic in Siberia, another group of scientists is doing the same thing, digging into what's known as yadoma, a kind of ancient frozen silt packed with ice. These deposits formed during the Ice Age, tens of thousands of years ago, and they're so dense with organic matter that they sometimes smell faintly of old plants when they thaw. At a place called Duvany Yar, near a small Siberian town named Chersky, researchers use rotary drills to take cores of frozen ground from cliffs that have been carved open by rivers and thawing permafrost. It's like taking the center of a loaf of bread and ignoring the crust. Only the untouched middle is trusted. The samples are sealed, labeled, and sent back to the lab under strict temperature control, just like in Greenland. The first rule in this kind of work is simple. The outside of the core is dirty. It's touched metal, air, and gloves, all potential sources of modern DNA. To make sure the inside remains pure, scientists literally shave away the outer layers with sterilized blades. Then they test for contamination using a clever trick. When they drill, they dust the bit with tiny glowing beads called fluorescent microspheres. Under a microscope, these beads shine bright. If any show up inside the sample later, it means modern material sneaked in, and that sample gets tossed out. It sounds extreme, but every step is designed to protect the integrity of what's inside. The teams drill dry, no fluids, no lubricants, because liquid could carry modern microbes into the hole. The reason this frozen soil is so valuable comes down to chemistry and physics. Arctic ground acts like a natural vault for DNA. Cold slows the chemical reactions that destroy molecules. Ice locks water in place, so it can't move and cause decay. And the minerals in the soil themselves actually grab onto DNA like Velcro, holding it tight for thousands, even millions of years. One mineral, a type of clay called smectite, is especially good at this. In experiments, smectite was found to bind about 200 times more DNA than quartz. That's great for preservation, but terrible when you want to get the DNA back out. When researchers tried to extract it, smectite only released about 5% of what it held. Other clays let go of maybe 10%. Quartz, on the other hand, released around 40%. That means the soil itself is both the guardian and the gatekeeper. It's kept the DNA safe for 2 million years, but it doesn't want to let go now. That's why scientists first test what minerals are present in each sample before they start extraction. They use X-ray diffraction to see how much smectite, quartz, and other clays are there, and then tweak their chemical recipes to coax out as much ancient DNA as possible. Back in Copenhagen, the Greenland samples go through an almost surgical process. Inside a clean, cold lab, researchers carefully split the cores and collect small scoops, sometimes just a few grams, from the inner section. They turn each tiny piece into a DNA library, which basically means isolating and indexing every genetic fragment they can find. From the 5 Greenland sites, they built 65 separate DNA libraries from 41 samples. Before running expensive sequencing tests, they do an initial check using something called droplet digital PCR, or dPCR. It's a high-precision method that divides a sample into thousands of microscopic droplets and asks a simple yes or no question. Is the target DNA here? Only then did they move to full-scale sequencing. They used high-end machines, the Illumina HiSec 4000 and NovaSec 6000, to read billions of short DNA fragments in parallel. Some libraries were even run through an enrichment process called Arctic paleochip, where scientists use custom-made molecular baits to fish for specific genetic material, like bits of mitochondrial DNA from ancient animals. These labs look more like clean rooms in a space agency than a university. The air is filtered, the rooms are sealed, and scientists work in full protective gear. Even the smallest mistake, a stray hair, a fingerprint, could contaminate a 2-million-year-old sample. Every run includes blank controls with no sample inside, to make sure the process itself isn't adding any modern DNA. Before any field team starts drilling, they also rely on decades of Arctic monitoring data to know exactly where to dig. Networks like CALM, the Circumpolar Active Layer Monitoring Program, track how deeply the Arctic ground thaws each summer. The data comes from sensors buried at over 100 sites across the polar region. The shallower the thaw, the better the chance the ground has stayed frozen for thousands of years, which means the DNA inside is older and better preserved. Another global network, called GTNP, records ground temperatures from boreholes across the Arctic. It helps identify which permafrost layers are still stable and which have started to warm. Scientists can even model this information using software like the GIPL model from the University of Alaska Fairbanks. It combines temperature data with snow cover, vegetation, and soil type to map out where the ground is most likely to stay permanently frozen. By overlaying that with satellite imagery, they can literally map harvest hotspots before ever heading out on snowmobiles. In places like Siberia, though, nature sometimes does the digging for them. As permafrost warms, massive slumps and river cuts reveal deep layers that have been sealed since the Ice Age. One of the most dramatic examples is the Batagay megaslump, a gigantic scar in the ground in eastern Siberia that has exposed layers dating back hundreds of thousands of years. Some teams have even drilled through frozen lake beds, pulling up cores over 60 feet long that capture both underwater and land-based sediments. No matter the source, the rule is always the same. Take only the untouched center, keep it frozen, and document every move. And now we reach the turning point, the question that started this whole obsession. We've seen how the soil is collected, protected, and decoded. We've seen the chemistry that keeps DNA intact for millions of years. But what did that ancient soil actually say once the scientists finally cracked the code? The oldest genetic story ever told. When the scientists finally decoded what was hidden in that frozen Greenland soil, they uncovered DNA revealing a Greenland that was once green, alive, and nothing like the frozen land we know today. What they discovered comes from something called environmental DNA, or eDNA. It's not taken from bones or leaves, but from the tiny genetic fragments every living thing leaves behind. When an animal moves, it sheds hair, skin, and waste. When a plant grows, its leaves and roots leave microscopic traces of DNA in the soil. Normally, those fragments break down fast, but in the Arctic, under the perfect mix of cold, dryness, and mineral-rich soil, those traces can survive for astonishing lengths of time, locked away like letters sealed in ice. That's exactly what happened at the Kapp-Kubenhavn Formation in far northern Greenland. Scientists pulled those microscopic clues out of the frozen ground and used them like fingerprints to rebuild a world that no longer exists. The shock wasn't just how far back the DNA reached, it was the world that it described. Before this, the oldest DNA ever recovered came from mammoth teeth in Siberia, roughly 1 million years old. The Greenland samples doubled that record. The fragments found in the Kapp-Kubenhavn sediments are around 2 million years old, confirmed by multiple methods. The geology of the site, the dating of the sediment layers, and the molecular age of the DNA all point to the same window, the early Pleistocene, when the world was much warmer and Greenland was something entirely different from the icy desert we know now. So what did those DNA fragments say about that lost world? Today, Greenland's far north is one of the harshest environments on Earth, a polar desert where only a few mosses and hardy shrubs cling to rock. But 2 million years ago, that same spot looked like a northern forest. The DNA reads like a roll call of trees and shrubs, poplars, birches, and cedar relatives known as thuja, mixed with willows, herbs, and grasses that normally grow hundreds of miles farther south. Out of the 175 kinds of vascular plants known in modern Greenland, scientists detected the DNA of 70 of them, plus many more that no longer grow there at all. In total, they identified over 100 plant genera, and here's the detail that stunned everyone. Almost 40% of them no longer exist anywhere in Greenland today. Instead, those same trees and bushes still thrive in the boreal and northern temperate forests of North America, places like Alaska, Canada, and the northern United States. That means Greenland's far north once hosted a full blend of Arctic and temperate life, an ecosystem that doesn't exist anywhere on Earth now. It wasn't a tundra with a few lonely trees. It was a true forest, dotted with shrubs and grasslands, supporting herds of big animals that needed real vegetation to survive. And the animal DNA backs that up. Hidden among the plant fragments were traces of mastodons, reindeer or caribou, hares, rodents, and geese, a community centered on plant eaters. Just imagine that for a moment. 2 million years ago, in a place now locked in darkness and snow for most of the year, there were forests alive with the footsteps of mastodons and the calls of birds nesting by rivers. You don't get herds of huge herbivores without trees to browse and shrubs to shelter in. That list of animals tells you the environment must have been rich, mixed, and full of life. Scientists call it a no modern analog ecosystem, meaning we don't have anything like it on the planet today. It wasn't the Arctic we know, and it wasn't a normal boreal forest either. It was something in between, a unique fusion of the two. Even the ocean left its mark. Mixed into those same layers of soil were traces of green algae and horseshoe crab DNA, species that live in warmer coastal waters. That fits perfectly with the bigger picture. 2 million years ago, rivers flowed across the land and emptied into shallow coastal seas. As those rivers eroded the land, they washed soil, leaves, and animal remains into the same sandy layers that built up at the shoreline. That's why the sediments contain both land and sea life, plants from the forests, mammals from the plains, and creatures from the nearby coast, all sealed together. The marine DNA provides the final clue. It means the water along Greenland's northern edge was warm enough for species that can't survive there now. If you want numbers to picture it, here's what the scientists calculated. At that latitude, the average summer temperature was about 50 degrees Fahrenheit, and the average winter minimum was around 1 degree. That's roughly 18 degrees warmer than it is there today. That difference might sound small, but it's massive in climate terms, enough to thin or even retreat parts of the Greenland ice sheet, turning ice into rivers and tundra into woodland. Cold-adapted species still lived there, but they shared space with warmth-loving trees and shrubs that had moved north as the climate allowed it. What makes this story so convincing isn't one bold claim. It's how many different lines of evidence line up to the same conclusion. The age of the sediments wasn't based on a guess or a single test. The layers of rock record Earth's magnetic reversals, those times when the planet's magnetic poles flipped. By comparing the magnetic signature in the soil with known reversals in the geologic record, scientists could place the deposit precisely in time. They also measured two rare isotopes, aluminum-26 and beryllium-10, which form when cosmic rays hit surface rocks. Once buried, those isotopes begin to decay at known rates, creating a second clock. Both of those clocks pointed to the same era, roughly 1.9 to 2.1 million years ago. Then there's the DNA's own internal clock. For certain plants, like birch, researchers compared the chloroplast DNA, that's the small genome inside a plant's photosynthetic engine, with modern relatives on its family tree. The DNA differences between them reveal how long it's been since they shared a common ancestor. The result gave a median age of around 1.3 million years, with a likely range between about 700,000 and 2 million years. That overlap fits perfectly with the geological evidence. So, you've got three clocks ticking in sync: the rocks, the isotopes, and the DNA, all pointing to the same ancient world. But skeptics had another question. How can we be sure this DNA isn't modern contamination? Maybe newer organisms crept in through cracks or lab mishandling. The team had an answer for that too. Old DNA carries scars. Over time, its molecular edges break down in specific ways, creating a chemical damage pattern. Using specialized software, scientists scanned each DNA fragment for those telltale marks. The Greenland samples showed exactly the kind of heavy damage expected from molecules that had been frozen for millions of years. And the team set strict thresholds. They only accepted species whose DNA showed strong evidence of ancient wear. If a sequence looked too clean, it was thrown out. To make sure the DNA picture wasn't lying, the team compared it with more traditional evidence, things like pollen grains and visible plant fragments found earlier in the same formation. The match was remarkable. The old pollen records already hinted at a green landscape, but they missed many species that don't leave much pollen or whose pollen travels poorly. The DNA added those missing names: trees, herbs, and shrubs that had never shown up before. When pollen, plant fossils, and DNA all told the same story, Confidence shot up. But not everything buried in the Arctic is just a memory. Some of it isn't dead at all. It's only been waiting for the right warmth to wake up. The frozen vault of living microbes. When the frozen ground of the Arctic begins to thaw, not everything inside it simply melts away. Some things wake up. Not dinosaurs, not sci-fi monsters, but something far smaller and more ancient. The microbes and viruses that have been sealed in the ice for tens of thousands of years, waiting in silence. In research labs, scientists sometimes take a tiny pinch of that frozen soil, warm it carefully, and add one special ingredient, a single-celled creature called acanthamoeba. It's a free-living amoeba that lives in fresh water, and in these experiments, it acts as bait. The goal is to see whether anything in the thawed soil can infect it. Most of the time, nothing happens, but sometimes, under the microscope, something stirs. In 2023, a joint French-Russian research team got an astonishing result. They managed to revive 13 different giant viruses from samples of Siberian permafrost and nearby frozen ground. Some of these viruses had been dormant for nearly 48,500 years, older than the last ice age. Each one successfully infected only amoeba, their chosen host. None could infect humans or animals. That's the crucial line. These are not human pathogens, not precursors to the next pandemic. They are proof of concept, evidence that certain viruses can survive the deep freeze of time and still replicate once they find the right host again. And yes, giant virus is a real scientific term. Under a microscope, these things are huge for viruses, so large that you can actually see their outlines using basic lab equipment. They belong to families with names like Pandoravirus, Pythovirus, and Megavirus, and some look like little jars or capsules sealed with cork-like tops. A typical flu virus is just a few dozen nanometers across. These giants can reach lengths of 80,000 nanometers. The 2023 study isolated new members from 5 major virus groups, all from just 9 soil samples. 7 pulled from ancient permafrost and 2 from frozen surface soils collected along places like the Lena River region and Kamchatka Peninsula. One of those samples even came tangled with mammoth hair. The scientists nicknamed some of the revived viruses accordingly: Megavirus Mammoth, Pandoravirus Mammoth, fitting names for something that literally shared a burial ground with Ice Age giants. And yet, viruses aren't the only ancient surprises the thaw can bring up. Sometimes, what emerges isn't even a living thing. It's information. Buried in the same frozen soils, scientists have found antibiotic resistance genes, or ARGS, the tiny genetic blueprints that bacteria use to defend themselves against antibiotics. If you've ever heard of vancomycin, one of the strongest hospital drugs we have, there's a gene cassette called VAN-A that lets bacteria shrug it off completely. About 10 years before anyone revived those amoeba-infecting viruses, a Canadian research team went digging in 30,000-year-old Beringian permafrost, looking specifically for antibiotic resistance genes. And they found them fully functional versions, including a VAN-A element identical in design to modern hospital strains. That discovery shattered the common myth that antibiotic resistance is something humans created through overuse of medicine. It's not new, it's ancient. Bacteria have been fighting chemical wars against one another for millions of years, using natural antibiotics, and the microbes under attack evolved defenses long before humans ever discovered penicillin. If the idea of ancient genes still working feels abstract, another experiment made it vivid. Researchers collected soil samples from the high Arctic, extracted random DNA fragments, and inserted them into harmless E. coli bacteria. Then, they hit those modified E. coli with modern antibiotics to see what would happen. Many of the ancient DNA fragments gave E. coli new powers of resistance, protection against drugs like penicillin, against tetracyclines, and against a hospital-grade antibiotic called amikacin. Some of those resistance fragments even worked across drug classes, offering cross-resistance to multiple compounds at once. That doesn't mean ancient ground will unleash superbugs when it thaws. Those genes are just the potential, the raw material. For any of them to matter, they'd have to jump into bacteria that infect humans, and then those bacteria would have to spread. That gene-swapping process, called horizontal gene transfer, happens naturally when the right microbes meet, but it requires the perfect mix of conditions. What these studies really show is that the Arctic permafrost holds a vast, ancient library of genetic tricks, a resistomy, and some of those tricks still work when scientists test them today. So far, we've seen ancient viruses that only infect amoeba, and antibiotic resistance genes that came long before humans ever made medicine. By themselves, those facts feel harmless, almost like something that belongs only in a lab. But the Arctic has already shown us what happens when biology in the ground crosses paths with modern life. Not in a petri dish, but out on the tundra itself. That lesson came in the summer of 2016, on the Yamal Peninsula in northwest Siberia. It was one of the hottest summers on record there, with air temperatures climbing into the mid-90s Fahrenheit, unheard of for that region. As the heat lingered, reindeer began dying in alarming numbers. At first, just a few, then dozens, then hundreds. By the end, over 2,000 reindeer had died across the open tundra. Herders started falling sick too. Doctors in the regional capital treated dozens of people, and the government ordered evacuations. A 12-year-old boy died. The cause turned out to be anthrax, the same bacterial disease once weaponized in labs and feared worldwide. The source, investigators concluded, was the thaw itself. The extreme heat softened permafrost that had been frozen solid for decades, exposing an old carcass or burial site infected with Bacillus anthracis, the bacterium that causes anthrax. When the ground opened, long dormant spores escaped back into the environment, and those spores are built to last. An anthrax spore is essentially a time capsule, a hardened shell with its metabolism shut down, waiting for the right warmth and moisture to come alive. In frozen ground, it can survive for decades, even centuries. The Yamal region hadn't seen an anthrax outbreak since the 1940s. Routine vaccination of reindeer herds had kept the disease in check for generations, but vaccination programs were dropped in the 2000s, and by the time that record heat wave hit, the safety net was gone. When anthrax resurfaced, it did so with brutal efficiency. Emergency vaccination campaigns began, quarantine zones went up, and carcasses were burned in deep pits to stop the spread. The scene was surreal. Hazmat suits against a blazing Arctic sun, helicopters ferrying vaccine doses to remote camps. It was a reminder that the Arctic doesn't just store ice and fossils, it stores history, including the biological kind. And when that history thaws, it can step directly back into the present. Thanks for watching. Now check out the videos popping up on screen for more unbelievable stories.